right, so that brings us to our last talk for today, and that is going to be a tag team effort from Joseph Crane and Peter Hoop. And so Joseph Crane is the co-owner of Jonah Ventures, which was founded in 2014 and helps clients answer ecological questions with DNA sequencing. And Peter has been with Swamp for 3 years and is a recent graduate of the Oregon State University College of Forestry. So they will be tag teaming this presentation on using Jonah Ventures for answering bioassessment research questions. Take it away.
Good morning. I'm Peter Haupt from the Swamp IQ unit. Next slide. To quickly review the SWAMP mission, we evaluate the physical, chemical, and biological quality of water to support a wide variety of users. Through carefully designed programs, we provide timely, high-quality data to assist other entities throughout the state. Next slide. In a 2020 program review, it was found that SWAMP's monitoring needs exceed their existing resources, and coordination between programs needs to be better integrated. This is where we think eDNA can provide great value to the program. Next. Metabarcoding can be implemented in a variety of ways and can drastically reduce collection costs, increase turnaround times, increase data quality, fill in data gaps like fish and other indicators, and improve coordination among programs using a single data type. Next. Metabarcoding gives us an opportunity to expand the scope of existing programs and provide a holistic panoramic view that incorporates a wide collection of species across the landscape, allowing us to examine the detail of a single species occurrence within a watershed as well as a greater assemblage at a single site. Next. Integrating soil, sediments, and biofilms can provide an even greater understanding of the ecosystem. Next. Commercially available sampling kits like those developed by Jonah Ventures, which you'll hear about in just a minute, provide simple standardized collection that can easily be implemented by volunteers involving the public in data collection in ways that could be complementary to established programs. These commercial services also provide accessible methods of data exploration as well as detailed spreadsheets for scientific analysis. Next. We're encouraging programs and regions to explore the benefits of eDNA and submit proposals to SWAMP. Next. SWAMP will continue to contribute to the molecular methods work group, developing protocols and standardizing data integration. We also hope to expand our partnerships and contribute to larger efforts like the U.S. Forest Service eDNA Atlas pictured here on the slide. You can see we're a bit behind our neighbors to the north and east. To this end, we've created a curated library, including journals and studies concerning field protocols, engaging the public, study design, and monitoring. This is available in a link I will post on the Slack channel. Next. The cost, efficiency, and comparability of eDNA data collection make it an ideal way to better coordinate between programs and allow our existing resources to meet our monitoring needs. Next. While the eDNA team was conducting research this spring, we were fortunate to be contacted by Nicole and Joe from Jonah Ventures and have been participating in their pilot project. Shuka and I have been using their sampling kits to sample from reference stations with historic data, and we'll be analyzing the results as they come in, but COVID and fires have slowed progress significantly. The kits are really easy to use and contain a preservative so that you can mail them in without any ice. This slide shows some of the Jonah Venture online tools with some of our sampling results, and you can see them at jonahdna.com. Here to tell you more about the project is Joseph Crane from Jonah Ventures.
Thanks, Peter. That was amazing that you got through that as quickly as you did. I had no doubts. Thanks everybody for listening for a couple minutes here. Just going to make sure that I can control screens. Potentially. All right, so just as a company, we've been around for 5 years or so. We process about 20,000 samples a year, and we've had over 350 clients that we've worked with. We'll do quantitative PCR as well as next generation sequencing, and we've got some new point of use tests that give you results back in 30 minutes that we're developing now. I'll talk about the next gen sequencing today. Essentially, probably the most important thing that we did is that we put everything in a box. So as far as all the sampling equipment is concerned, it's a syringe, there's a syringe filter, preservative, caps, gloves, mail-in envelope. We can get all that into a box. Anybody can use it. That's probably been the most important thing that we've done, even though it seems like the simplest. From there, we've paired that with smartphone apps on iOS and Android so people can easily record their metadata, real basic metadata, which can be expanded at a later point. It's all going onto the cloud, into a non-relational database that can hold a lot of different data. And then that's really all an individual has to do is be able to suck up water, push it through a filter, mail it in, record some metadata. Then we'll do all the hard stuff as far as extraction of DNA is concerned, amplification, and sequencing, bioinformatics. Really briefly, we've had samples come in from all over the US. We're in almost all 50 states right now. This is from a couple of weeks ago. But once you get your data back, all of that is accessible through the data portal, as well as we'll send out files to people too. So we've got some experience in a lot of different environments, not just California. And the real simplified version of what we present for fish is going to be a list of presence of fish species. We've got more detailed data that we send individuals, but on the app, or excuse me, on the data portal, you'll see that. And then we'll include images of the fish, its distribution as far as what's known, and then some descriptions for citizen scientists. It's helpful for them to learn about what they're finding. The success of this is I've been pleased with on the fish side of things, as well as phytoplankton. Just looking at the fish data, this is from a single site. These are the presence of roughly 25 species of fish that we found in South Dakota, just from 100 mils of water. It works pretty well. How many replicates we need, for example, to get everything that you would find is still open to exploration, but we're able to get a pretty high diversity from just 100 mils of water pretty regularly. And what we're finding in different places matches up with what we are known to be in different places too. So this would be a South Dakota site. And then if we put it together for Alaska, for example, we're finding Alaskan species. It's pretty consistent. So I think we've got this platform relatively well worked out. These are just the fish data here. We're doing it for phytoplankton relatively regularly with just as high a success rate, and are working on other primer sets to look at different organisms also. Where we feel like this will have a rapid impact and a pretty major impact is, just like Peter said, is through bioassessment, and that's what we've really been concentrating on. Whereas with traditional techniques of collection, sorting for macroinvertebrates, for example, painstaking identification, generation of IBIs, it's expensive, it's slow, it doesn't give us good resolution taxonomically as well as for sites. But once we start to use eDNA, whether that's sucking up water or taking macroinvertebrates and putting them in a blender and just sequencing it, it's less expensive, it's a lot faster, and it seems like we're getting much more accurate water quality indices. So we think that bioassessment is going to move to eDNA at some point, and we're hoping to speed that up as fast as possible. And even right now, just with the fish data that we have on presence absence, we can use the data that we have for coefficients that represent water quality and show differences in water quality between streams that we find in New Hampshire, Alaska, and Oklahoma, just based on scores that other people have assembled for those fish species. Getting to IBIs and more sophisticated IBIs with eDNA is something down the line, but we're getting pretty close. That was my last slide, so hopefully I finished under time, and that'll give us time for questions. Thank you, Peter, for introducing everything.
Okay, that was great. So it looks like we have about 10 minutes that we can open up as a bit of a Q&A session for anyone that was in this eDNA session. So everyone's welcome to enter their questions onto the Slack channel. And I would invite all the speakers in this session, you can unmute yourselves And as we wait for questions to come in on the Slack, I had one question I was going to ask Joseph and Peter. Maybe there's a question for Joseph. Can you talk a little bit about how you're handling on that last slide that you were showing, calculating fish IBIs? How does it handle abundances or relative abundances if you're working with meta barcode data and you're looking at calculating IBI scores? I think that's a question that a lot of folks would have.
Yeah, it's what we found empirically is that relative abundance for fish from this approach of metabarcoding is probably not that reliable, not yet at least. And so we're just working off of presence absence. And you might be able to get relative abundance from having replicates, but from an individual sample, it's just a list of presence. For phytoplankton, relative abundance is a lot more possible because there's a lot more DNA in there. And so we feel like getting more accurate IBIs from phytoplankton is probably going to be, and again, we know that different organisms provide different signals, but it's probably going to be the future more than fish. But at the same time, we can get fish, macroinvertebrates, phytoplankton all in the same fell swoop. And so more than likely, those IBIs will work across all those organism groups. Thanks, Susie.
Awesome. Okay, we did have one question that just came in that asks about local eDNA libraries. So are those needed in small headwater streams? Do you need a certain number of individual species to get a good ID of species? I participated in a study at OSU. This is from Cajun James. We had trouble with low flow numbers of species. So let's take that question in two parts. First off, I'd love to hear from any of you all on what about local eDNA libraries and if they are needed. Do we have any strong feelings about DNA libraries? I know I do, but feel free to speak up.
If I can jump in, because we've worked on this pretty extensively. Right now, it's not just local libraries, it's global libraries. There's somewhere on the order of 1,200 fish species in the US, and roughly a third of those have been sequenced. That's it. So we don't need necessarily local libraries, we just need the ones that have each species once. And on the phytoplankton side, it's even worse than that. It's probably 5% of the species have been sequenced. So we get pretty close, but even California with its reduced number of fish species, for example, that are there, there's still a whole bunch that haven't been sequenced yet. So yeah, we've got a ways to go just to get to the first pass. I don't know if other people have opinions on that.
Yeah, I can speak to that too. That's something that that my lab at UC Davis is involved in. And my PI, Andrea Schreier, received a Prop 1 grant to actually put together reference libraries for the San Francisco estuary targeted at fish and larger macroinvertebrates. And we see that as a really important part of moving eDNA into a place where it can be more useful to management.
Yeah, another data set that I know of is developed by my colleague Zach Gold called FishCar. So it's a marine fish 12S California specific reference database. So I think it's definitely like very important to construct local reference DNA derived libraries if you see that you have trouble finding the number of species or you have trouble like finalizing, or for example, if you it's possible that you found match to like family level for like a different species at a different locality. That's like where it's necessary to have those.
Okay, awesome. I just got another question that came in that says, can you get terrestrial plants in aquatic eDNA? Is there anyone that wants to take that question?
Yes, you can. And one source we think is through pollen, but I think it's also possible to detect plants through detritus or just bits of material. But yeah, definitely.
Yeah, I think the idea of the autochthonous versus allochthonous input of eDNA is a big one, an active area of research, and a lot of folks are interested what can be getting washed into your watershed if you're looking at aquatic eDNA. So I think that's an active area of research. But the short answer is yes, you can get terrestrial DNA in your aquatic samples. Any other comments on that?
I'll just add to that we could take that as far as terrestrial animals or mammals as well, especially in areas where maybe there's not frequent water sources for those mammals, and we know that they probably are visiting a certain aquatic area often.
Awesome point. All right. So one of the other questions that I was hoping that we would hear from the panelists about are what's the area that you would like to see the greatest amount of research efforts focused on in the next 5 to 10 years? So maybe we can just go through everyone. If you have strong feelings on where you think our eDNA field really needs to concentrate its research efforts right now to help advance eDNA and the integration into routine monitoring efforts. I can start off with my opinions. I think what we heard a lot today already was that some of the DNA reference libraries can really be constraining our ability to identify some key target taxa in our samples. I think to a certain extent that's some low-hanging fruit. We know we're good at sequencing reference sequences. It's just a matter of devoting the time, effort, energy, dollars, and hopefully being able to actually sample those organisms that we need those reference sequences for. So I would love to see some really big progress in that arena so that it's just something we don't have to worry about in 5 years. Anyone else?
Yeah, I'll add that I'd really like to see more data on using eDNA for abundance estimates, which we're already seeing a lot of publications on this, but I think in the next 5 to 10 years, we're going to really refine our ability to use eDNA to create these abundance estimates. And then also looking at doing like RNA-seq or getting at gene expression ultimately from a water sample potentially, and how that might have implications for ecosystem condition and how gene expression might be changing, especially for thinking in California, post-drought, post-wildfire, things like that.
That would be so great. That would be such an exciting area to see some more development. We talk a lot about it in the harmful algae world. If we can get at RNA transcription as an early warning indicator, so that would be awesome. Anyone else?
I think your study on fate and transport is really important, that, you know, we can sort of determine how far this is moving. I thought the studies today were great.
Awesome. Yeah, exactly, Peter. I think that's something a lot of folks would love to see. And in the increasingly more complicated arenas also, like estuaries and beyond, where we're dealing with our tidal regimes. That would be awesome. Anne or Meiji?
I would think that SOPs is the most important thing, like next steps. And maybe also trying to, like, we do a lot of experiments to test like the transportation distances, to test like the replicates and the stochasticity in them. I think it would be important if we could model them to some extent so that we don't have to completely use empirical data to inform our choices.
Awesome.
Some things that I see are on the communication front. So communicating not only the potential, but also the limitations. eDNA is, you know, it's another approach, and it, like all methods, it has some limitations. And I think that will help integrate eDNA with other methods where it can be complementary.
Yeah. Awesome. Awesome. All right, I see another question that came in that is from Keith, and he asks, have people gotten eDNA detections at distances over 1,000 meters? What is the maximum spatial scale people think eDNA can be detected in aquatic habitats? Does anyone want to take that one?
Yes, I've seen those numbers published, and I think it's very possible. I think it's probably varies a lot on habitat and species. You know, how abundant was the species to start out with? How does the transport work? But yeah, there's a lot of really good questions to explore in those long distance transports.
Yeah, something that comes to mind thinking about long distance transport is a study done by Christy Diner that they looked at distance of travel of, I want to say, an invertebrate species, maybe Daphnia, that was transported from like a lake outlet downstream up to 10 miles in this stream. So possibly really long distances, but for fish studies, it doesn't seem like we've shown that that we have that long of a distance necessarily.
Yeah, I think across the board, we're seeing there are so many different environmental parameters that go into dictating how far that eDNA can travel and how long it persists in the environment. So I'm really excited to see hopefully in the next 5-10 years, our ability to constrain some of those estimates so that we have, some great models that we can plug some numbers into and get a good estimate for our target environments on how long we expect that DNA to exist. And that'll help us determine our sample schemes for years going forward. So with that, I think we're at time.